Hello, this is Dr. Nasheen Ahmed from the University of Kansas Cancer Center presenting on CAR T in multiple myeloma. These are my disclosures. Our learning objectives today is to identify eligible relapse refractory multiple myeloma patients and understand current indications. Second objectives is to look at efficacy and safety of the approved therapies. And lastly, we will discuss the future and clinical trial landscape in this field. Historical context. We have a publication in 2019 which showed that penta-refractory patients have a poor prognosis with a survival of less than 6 months. Now, since then, we have had several advances, especially with the identification of BCMA as a target on plasma cells. BCMA is an ideal target because it's expressed in almost 100% of the plasma cells and is not expressed on other cell surfaces, and therefore the off-target effects are minimal. Both of the currently approved CAR T products in relapse refractory myeloma target BCMA. The first approved product in 2021 was idecabtagene reclusil, ide-cel, and the second one, which was approved in 2022, was ciltacabtagene autoleucel, also known as cilta-cel. Both were approved for a similar indication for patients with relapse refractory myeloma who had progressed after 4 or more lines of therapy and had to have prior IMiD, proteasome inhibitor, and CD38 monoclonal antibody exposure. Now in 2024 onwards, we will discuss anticipated approvals in earlier lines. This is a cartoon showing some differences in the constructs of the CAR T receptors for ide-cel and cilta-cel. While they are generally similar and have similar co-stimulatory domains which stimulate and activate the T cell, cilta-cel expresses a single domain antibodies directed against 2 distinct epitopes of the BCMA target antigen as opposed to ide-cel that has 1. Discuss the pivotal trials that led to the approvals of these 2 novel therapies, specifically the KarMMa-1 trial for ide-cel and the CARTITUDE-1 trial for cilta-cel. Both of these were single-arm trials involving leukapheresis bridging followed by lymphodepletion with fludarabine and cyclophosphamide, and then the CAR T infusion. For KarMMa-1, eligible patients were those with myeloma who had at least 3 lines of therapy, exposed to IMiD, proteasome inhibitor, and anti-CD38 monoclonal antibody. And all patients had to be refractory to the last therapy. Different dose levels were evaluated in KarMMa-1. The CARTITUDE-1 trial for cilta-cel was a similar study design, a phase 1b/2 study with a fixed target dose of 0.7 million CAR T per kilograms. All patients in the study had relapse refractory myeloma and at least 3 lines of therapy with previous exposure to an IMiD, proteasome inhibitor, and anti-CD38 monoclonal antibody, or were double refractory to PI and IMiD. Here are the baseline characteristics for KarMMa-1 and CARTITUDE-1. Again, there's no direct comparison, but numerically it appears that age and median lines of therapy as well as triple refractory status was similar for both of these trials. And in the KarMMa-1 trial with ide-cel, there seem to be more patients with high-risk cytogenetics and extramedullary disease, while there were more penta-refractory patients in the CARTITUDE-1 study. Again, this is not a head-to-head comparison. Some outcomes for ide-cel and KarMMa-1. The overall response rate was 73% and 81% for the target dose, with a complete response rate or better being 33%, 39% for the target dose. Now, this includes responses in all subgroups, including high-risk cytogenetics, extramedullary disease, high tumor burden, and RISS-3. With a 24-month follow-up, the median PFS was 8.8 months and 12.2 months for the target dose, and the median duration of response was 10.9 months, which was actually the median duration of response was not reached for those with complete response or better. The median overall survival was 24.8 months, and the estimated 24-month overall survival rate was 51%. CARTITUDE-1 For cilta-cel, results are shown here. The overall response rate was 98%, with 83% being at stringent complete response at 2 years. The 12-month PFS was 77% and 18-month PFS was 65%, with a median progression-free survival not reached. Overall survival at 18 months was 81%. Median overall survival and median duration of response were not reached. I will highlight some of the adverse effects and toxicities in the two pivotal trials we discussed. Cytokine release syndrome was seen in the majority of patients who got ide-cel or cilta-cel, with high-grade CRS being 5% or less. The onset to CRS was shorter for ide-cel at 1 day from infusion compared to 7 days for cilta-cel. Immune effector cell-associated neurotoxicity syndrome, or ICANS, was seen in around 17 to 18% of patients with cilta-cel and ide-cel, and high-grade ICANS was seen in less than 5% of patients with either ide-cel or cilta-cel. The onset to ICANS was again shorter in patients who received ide-cel at 2 days compared to cilta-cel, where the onset was at a median of 8 days. Interestingly, the CARTITUDE-1 trial reports other neurotoxicity, movement disorders such as Parkinsonism, in 12% of patients, while there is none reported with ide-cel. Prolonged cytopenia at 1 month was in around a third of patients in both the KarMMa-1 trial as well as the CARTITUDE-1 trial. And infections of any grade were seen in the majority of patients on the KarMMa-1 trial as well as on CARTITUDE-1 trial, with high-grade infections or severe infections being around a fourth of patients in either study. So presently, we have two products, ide-cel and cilta-cel, that we have just discussed that are both approved in patients who have progressed after 4 lines of therapy. There are other advances that are anticipated, such as approval in earlier lines, novel targets, GPRC5D CAR-T and other targets, new manufacturing platforms for shorter manufacturing and more efficient manufacturing of CAR-T, as well as allogeneic products. So let's discuss some of these. Let's first talk about the anticipated earlier approvals. We have the KarMMa-3 trial for ide-cel, which is a phase 3 trial that enrolled patients who had 2 to 4 prior lines of therapy, were triple exposed and refractory to last therapy. There was a 2-to-1 randomization to either ide-cel or standard of care, and standard of care is shown here as patients who got these regimens. The table in the middle, in the center, shows the responses, which clearly favors ide-cel over the standard of care. The overall response rates was 71%, with over half of those responses being complete responses or better. CR rate was 39%, which is almost 8 times that of the CR rate in the standard of care arm. The median duration of response was 14.8 months, higher than that of the standard of care. MRD negativity was 20% in the ide-cel group versus 1% in the standard of care group at 3 months, and the median PFS was 13.3 months, triple that of the standard of care. The FDA Oncological Drug Advisory Committee on March 15, 2024 voted in favor of moving ide-cel to earlier lines of therapy, and now the final approval decision by the FDA is eagerly awaited. Cilta-cel was studied in CARTITUDE-4 for earlier indication as well. This trial studied cilta-cel versus standard of care, which was DPD or PVD. The patients enrolled had 1 to 3 prior lines of therapy, and all were lenalidomide refractory. There was a 1-to-1 randomization, and median line of therapy was 2 lines of therapy. The responses are shown in the table in the center. The overall response rate was 99.4% in those who received cilta-cel and 67.3% in the intention to treat population. The intention to treat population includes those who were leukapheresed and did not get the CAR infusion. Most of the responses were CR or better. The CR rate was 86%, which was 4 times higher than the standard of care. Median PFS and duration of response was not reached. The MRD negativity rate was 87.5% versus 32.7%. And the 12-month PFS was 76%, which was higher than the standard of care arm. The overall survival data continues to mature. The mortality was similar in both groups. Based on the results of these trials, the FDA Oncological Drug Advisory Committee on March 15 voted in favor of moving cilta-cel to earlier lines of therapy, and now the final approval decision is also awaited by the FDA. Than earlier approvals, there are also other trials expected to improve on the outcomes with presently approved therapies. I will highlight just a few. Here in this slide, we have a novel agent, the JSMD194, which is a gamma secretase inhibitor. The gamma secretase inhibitor prevents cleavage of BCMA from the surface of the plasma cells and increases the density of BCMA on the plasma cells, thereby increasing potentially the efficacy and binding of BCMA-directed CAR T. So combinations of the CAR T with this product is being evaluated in a clinical trial. Other combinations and sequencing strategies, especially with the BCMA and GPRC5D bispecifics, such as teclistamab and talquetamab, are also being evaluated in clinical trials. Moreover, novel targets for CARs are in development, such as those targeting GPRC5D and FcRH5. Example, MCAR-H109 is a GPRC5D targeting CAR T, and shown here is a patient with a plasmacytoma of the chest wall, which responded beautifully to treatment with this CAR T. And this was in a study of 19 patients in the phase 1 study using this product. Other important efforts are directed towards the CAR T manufacturing platform. For the autologous products, that means improving manufacturing time and predictability. There's also other platforms looking at allogeneic CAR T cells, which are T cells that are not derived from the patient, but rather from a third party, such as a healthy donor or similar source. The allogeneic platform has advantages such as the product being off the shelf and readily available. There are many studies with allogeneic CAR T ongoing, all with unique designing to mitigate the risk of graft versus host disease and rejection. So in summary, some take-home points. Any patient with relapse refractory multiple myeloma who has had 4 or more lines of therapy should be considered for CAR T therapy. There are 2 presently approved products, which are ide-cel and cilta-cel. Some of the efficacy and safety data we have discussed, and toxicity specifically with regards to cytokine release syndrome, neurotoxicity, prolonged cytopenia, and risk of infections. There are other studies ongoing evaluating the real-world toxicity and outcomes with CAR T therapy. As for the future, we are looking and anticipating earlier lines approvals of ide-cel and cilta-cel based on the KarMMa-3 data and the CARTITUDE-4 data. Novel constructs, novel targets, improvement in manufacturing platform, and combinations with small molecules as well as sequencing with bispecifics. Thank you.